Okay, let's dive into this. Welcome to today's Explainer, where we're unpacking one of the absolutely wildest scientific and financial stories of our modern era. When most of us think about the mRNA vaccines rolled out during the 2020 global crisis, we picture this ultimate overnight success story, right? Like it was this sudden, miraculous scientific breakthrough born completely out of the sheer chaos and desperation of that specific year. Well, actually, scratch that. That's a total myth. The real story? It's a decades-long saga. We're talking immense financial risk, unsung scientific pioneers grinding away in the lab, and literally massive biological hurdles. We're going to break down exactly how this tech came to be, the mind-boggling wealth it generated for a select few, and of course, the incredible science that makes the whole shebang work.
To really grasp the scale of what we're talking about here, just consider this staggering number: $1.8 billion. By May of 2020, that was the estimated net worth of Moderna's CEO, Stefan Bancel. Just wrap your head around that for a second. While the world was literally locking down and the vaccine race was kicking into high gear, the sheer financial windfall for the folks steering these companies was just astronomical. Moderna's shares more than quadrupled that year, and that was all driven by early phase 1 trials showing their experimental vaccine actually created an immune system response in humans.
And this billionaire boom wasn't just limited to the CEO. When Moderna's shares surged 20%, hitting a record $80 a pop, following those encouraging early trials, a very specific group of early investors cashed in big time. So alongside Bancel's $1.8 billion slice, you've got Harvard professor Timothy Springer, whose holdings topped $1 billion. And right next to him is Bob Langer, an MIT professor and board member. His 3.2% holdings surged to nearly $935 million. That basically rounds him up to being the firm's third billionaire shareholder. It's an incredible financial climax. But it begs a pretty fundamental question.
Section 1. The magic of mRNA. How is this record-breaking medical timeline even possible?
We just talked about the massive financial outcome in 2020. But to truly understand this story, we have to pivot away from the stock market and look closely at the underlying science. What exactly is this tech that let these pharma companies move at a speed the industry had never seen before? Well, it all comes down to what mRNA actually is. So the crucial point is that messenger RNA is basically just an instruction set. It's a short transcript of a much longer DNA code, and its one and only job is to carry genetic instructions straight to your cells. But here is the absolute kicker, and why this technology is so revolutionary and safe. As a messenger molecule, it does not alter your body's own genetic code. It never breaches the cellular vault. It simply drops off the instructions and leaves.
Let's walk through this super elegant, three-step biological sequence. Step 1, the synthetic mRNA gets injected into the body. Step 2, it acts like a manual, instructing your cells to code for copies of a certain protein. Now, in the case of COVID-19, that was the famous spike protein you see on the outside of the coronavirus. And finally, step 3, your body recognizes this protein as a foreign invader and starts pumping out neutralizing antibodies. It is a completely revolutionary approach. Because they only needed the genetic code to write these instructions, it meant a vaccine could be formulated mere days, yes, days, after the virus's genetic code was mapped and made available. The rest of the time, that was just spent on the rigorous clinical trials.
Section 2. Pioneers behind the tech. Let's look at the unsung scientists behind this.
Because look, writing those instructions successfully took decades of trial, error, and frankly, a lot of frustration. Let's rewind the tape way past the chaos of 2020. You simply cannot talk about mRNA without enthusiastically introducing these two foundational pioneers, Katalin Karikó from the University of Pennsylvania and her collaborator Drew Weissman, an immunologist from Boston University. Long before the pandemic, way before the billions of dollars started flowing, these two laid the essential groundwork for using synthetic mRNA. They tackled the super early fundamental problems of just getting the body to accept synthetic mRNA without instantly destroying it. Their painstaking research in the lab, that is the bedrock upon which modern biotech applications were built.
And this brilliantly illustrates the next major leap in our timeline, a massive 2010 breakthrough at Harvard by a researcher named Derek Rossi. Check out the old method on the left. Before Rossi, scientists were trying to reprogram human cells, like turning adult skin cells into stem cells, using an old method that relied on viruses to insert genes directly into the cell's DNA. Honestly, it was dangerous. It carried a real risk of triggering cancers, it altered the cell's properties, and it was terribly inefficient, only working about 0.001 to 0.01% of the time. Now, compare that to Rossi's mRNA method on the right. He used synthetic messenger RNA to reprogram the cells. The result? It was completely non-integrative, meaning it was safe for patients because it didn't breach the DNA. And astonishingly, it was orders of magnitude more efficient, working 1 to 4% of the time. A real game changer.
Rossi himself summed up the magnitude of this perfectly. Read this quote with me. The method does not in any way breach genomic integrity, and it gives us a way to directly program and direct the development of the iPS cells, which are those reprogrammed stem cells we were just talking about. See, the huge problem Rossi solved was the body's natural defense system. Normally, if you introduce foreign RNA into a cell, the cell panics. It thinks it's being infected by an RNA virus, and it retaliates with a massive interferon response, basically committing altruistic suicide to stop the virus from spreading. But Rossi successfully modified the RNA so it wouldn't set off that antiviral alarm system. He tricked the cell into safely accepting the instructions, allowing scientists to express proteins in a cell for days and even weeks without any nasty adverse reaction.
Section 3. Rise of Biotech Giants. From the labs straight to the boardrooms.
It was exactly these painstaking lab breakthroughs that birthed the multi-billion-dollar companies we all know today. Once the foundational science was proven safe and efficient, the race was on to commercialize it. Let's move to the timeline and see how this builds, completely shattering the illusion that mRNA tech was some sudden, overnight success. Okay, in 2006, Japanese researcher Shinya Yamanaka discovers the old viral method of cell reprogramming. Just 2 years later, in 2008, BioNTech starts operating from a small lab in Mainz, Germany. Then, 2010, Derek Rossi achieves his safe mRNA breakthrough at Harvard, and that very same year, a group of investors founds Moderna Therapeutics, later known just as Moderna, over in Massachusetts. And then, it took another 10 full years of research, investment, and development before the first mRNA COVID-19 vaccines were approved in 2020. That is a 14-year march from Yamanaka's initial discovery to the actual vaccine rollout.
Which brings us right back to the extreme risk of those early days. Remember Timothy Springer from our billionaire breakdown earlier? Well, back in 2010, when Moderna was just being founded around this unproven, highly experimental mRNA technology, Springer made a $5 million gamble as a founding investor. I mean, he put his own money on the line for a technology that many thought would never, ever work safely in humans. A decade later, that single unproven stake ballooned to $1 billion. It neatly ties our decades-long timeline right back to the staggering financial climax we started this explainer with.
And that leaves us looking toward the horizon. Because the true story of mRNA, it's not just about a single vaccine or a single chaotic year. Rossi's breakthrough proved we have a safe way to transiently express any protein in a cell, all without triggering an antiviral response. The applications for things like regenerative medicine and curing protein deficiencies are truly boundless. So, the ultimate question to leave you with today is this. With COVID-19 conquered, what other incurable diseases will mRNA be programmed to defeat next? The biological instruction manual has been unlocked. Now it's really just a matter of what code we decide to write. Thank you so much for joining me for this explainer, and as always, keep learning.